specifically recognize some sequences in the genome at cis-regulatory regions, and basically by binding really schematically to the genome, they switch on and off the transcription of genes. And so when you want to analyze that, the first thing, one thing that you want to do, of course, is to predict as accurately as possible the precise locations of the transcription factor binding sites. So classically, what's done to model that computationally is to use position frequency matrices, which basically summarize the motif, so the binding sites which are known to be the transcription factor binding sites recognized by your transcription factor of interest. Okay, so basically you have the frequency of having ACG or T at each one of the positions within your transcription factor binding sites, within the motifs that is recognized by your transcription factor of interest. And then you can derive from these frequencies a position weight matrix with basic log transformation in a way in order to use then this model to predict transcription factor binding sites on the DNA sequence. And one big assumption that this model is doing is assuming the total independence between the positions at the transcription factor binding site. And it has been recurrently shown not always to be the case. And so 2 years ago, we published the transcription factor flexible models, which are capable of capturing the successive dinucleotide dependencies within transcription factor binding sites. So basically what we're doing is using a first-order Eden-Markov model to capture these dependencies, where basically here at each one of the position represented by a state of your transcription factor binding site, you have the probability of observing ACG or T, which is dependent on whether you observed ACG or T at the previous position. And then you can learn the parameters of the Eden-Markov model using ChIP-seq data and use this model then to scan any DNA sequence in order to predict transcription factor binding sites. So basically here, what we have is a model that is capable of capturing the sequence information at transcription factor binding sites, which are recognized by a transcription factor of interest. But you have different layers that can be recognized by the transcription factor, for instance, the structure of the DNA or the shape of the DNA. And recently, Dr. Remo Roos and his team at the University of Southern California developed a tool called DNA Shape, which enables for the prediction of the value of minor groove widths, roll, propeller twist, and helix twist at every position within a DNA sequence. And they've used this tool to develop the genome browser called GB Shape, where you can have access to these values, these DNA shape features, at every single position within your genome of interest, for instance, the human genome. And so the next step that you can think about is, of course, to combine the sequence information that is captured, for instance, by the TFFMs with the structure information or the shape, DNA shape information that is captured by these different DNA shape features. And so there have been multiple studies already that looked into that in order to combine the sequence and the shape information to model transcription factor binding sites. Here I highlight some of the recent publication this year, where basically they've shown that the incorporation of DNA shape was enabling for better predicting or better modeling transcription factor binding sites using in vitro data. And in our study, what we wanted to do is to focus on in vivo data to see how these translate to ChIP-seq data. So the idea was to address the following aims, where first we wanted to be able to construct a computational model that is capable to combine both DNA sequence and shape features in a single model, and that you can train on in vivo data on ChIP-seq experiments, and then show that then you can use this model to predict transcription factor binding sites, to discriminate ChIP-seq bond regions from genomic background, and improve the prediction on these in vivo data. And then finally, if it's the case, the idea was to look at whether the incorporation of DNA shape was improving the prediction of TFBSs for all the transcription factors or only for a subset of them, which DNA binding domain was enriched in the ones that are improving a lot, and so on. And finally, to analyze what were the DNA shape features that were the most important, and whether this importance was the same over all the positions within a transcription factor binding site or specific to some positions. So to do that, this is what we've done. So we used an ensemble machine learning approach, namely a gradient boosting based on decision trees, where you represent a potential transcription factor binding site as follows. So here is a potential TFBS. You have the sequence here. So we compute the score that you can derive from the transcription factor flexible models that I call here a hit score in the vector. And then I compute this vector by adding up all the values of minor groove reads, propeller twist, roll, and helix twist at every single position within this sequence. And this characterize this potential transcription factor binding site. And then you can learn your model, you can learn the parameters of your classifier, trying to discriminate ChIP-seq bond regions from genomic background and see how it performs. So we've applied that to a 400 human ENCODE ChIP-seq data sets, and we've computed using a tenfold cross-validation approach the area under the receiver operating curve. And here you have the accuracy that you obtain. So here on the x-axis, you have the accuracy that you obtain when you use the TFFM score only to make this discrimination. And then here you have the accuracy, basically, that you obtain by using the DNA shape information into the classifier. And so what you can see is that all the points basically are here on this upper left-hand side, showing that when you use DNA shape information, you increase your the predictive power of your model for discriminating ChIP-seq bond regions from genomic background. And here what I've done is looking at the difference here in terms of accuracy by obtaining this area minus the area that you get with the TFFMs, and you have that for all the data sets where I ranked these with respect to this difference. And you see here that there is a subset of the ChIP-seq experiments for which we observe a very strong improvement in the predictive power of the model. And so we were wondering whether there were some specific transcription factors, some specific TF families that were enriched within this region. And so here you have the same plot, and at the bottom here you have the different transcription factor families that have been used in this study, and you have the points here corresponding to the data set associated to this TF family, which are located with respect to their location here on this x-axis. And we see that there is an enrichment here on the right-hand side for some specific transcription factor families. We compute the p-value of this enrichment using a Mann-Whitney U-test, one-sided, and then we correct for multiple hypothesis testing. And what we observe is that there is a strong enrichment for E2F and MATS domain transcription factor families to be located here on the right-hand side, showing that for these TF families, we observe a very strong improvement in our capacity of discriminating ChIP-seq bond regions from genomic background when we incorporate DNA shape information. And we validated that a little bit further by looking at a totally independent data set where we combined multiple MATS domain transcription factor ChIP-seq experiment from plants for 7 of them. And we've done the same analysis where you have here the area under the receiver operating curve obtained with the TFFMs and with the classifier combining TFFM score with DNA shape information. And we see that for most of them there is a very strong improvement in predictive power when we incorporate the DNA shape information here, going from 60% accuracy to almost 100% accuracy. So this shows that the incorporation of the DNA shape features significantly improved the TFBS prediction for those MATS transcription factors. And then we looked at what were the properties, the DNA shape features that were the most important for this discrimination. And so you have here the results. Here you have the area under the receiving curves for each one of the data set when you use the 4 DNA shape features or only one of them at a time. And what we observe is that for all of them, the propeller twist is really the most important DNA shape feature for discriminating ChIP-seq bond regions from genomic background. And then when we look at the importance of each one of the features, depending also on the position within the transcription factor binding site, here you have the AGL15 example. You have the heat map showing the feature importance that you can derive directly from the models. And you see here the darker, the most important, that the propeller twists at some specific positions are really critical for modeling the TF binding specificity of the MATS domain TF. So to conclude, our analysis represents the in vivo counterparts of all the in vitro analyses that have been performed so far. And we've showed that we can construct directly from in vivo data, from ChIP-seq experiment, a model combining both DNA sequence and shape information to improve the prediction of transcription factor binding sites in these in vivo data. And when we look at TF families, we see that the incorporation of DNA shape information is most beneficial when you look at specific TF families. From our data set, we see the enrichment for E2F and MATS domain TF families. And finally, when we focus on MAT domains, we see that the propeller twist is the most important feature, and which is also a position specific. And finally, of course, I would like to acknowledge my collaborators on this work, Wei Thwasserman, my supervisor, Dr. Remo Rose, who is in the room, Lin Yang and Supei Chiu, and also Francois Auriol and Julie, and of course the fundings. And I would like to thank you very much, and I would be happy to take any questions. Thank you.
Yeah, thank you. I'm actually a bit, well, I'm actually a bit more interested in the other side of your curve. So could you comment a bit on the type of transcription factors that don't really benefit that much from including shape information? Did you see any patterns there?
So I didn't specifically look into those ones. We have a lot of ChIP-seq experiments that come from CTCF, and CTCF I don't see a strong enrichment. So that's really clear for this one. For the other ones, I would have to look into that. I didn't really look into what were the families that were not really benefiting from the DNA shape. Overall, we see that it improves, but for some of them it's really incremental. But I didn't do the analysis the other way around. Yeah.
Hi, very nice talk.
Thank you.
So you showed that the shape features matter for improving the prediction. To what extent is this claim dependent on the score used to make the predictions? In other words, to go from the motif model to the score that you then correlate with or use to predict the peaks. If that score, the regression or classification that uses the motif to assign scores to peaks.
So you mean the correlation with the peak score per se?
That's right. Whatever you use to compute a score from the motif that you then compare to the chip data, that scoring function is not sacrosanct. There's a lot of research needed on that scoring function itself, and therefore, to me, the claim is largely dependent on the scoring function as it stands.
Okay, so I didn't look at the specific value of the peak score, so I didn't look into that. The idea was really to make a discrimination whether it's binding or not, basically. So of course it can be dependent on how you call the peaks. I agree with that.
No, sorry, I didn't realize. So you're doing a binary classification.
Yes.
In that case, the classifier is the scoring function for me.
Yes.
The thing that uses the motifs, that
yes.
is intrinsically tied to the claim.
But I mean, so you mean the score that I use, the TFFMs, for instance?
That's
that
right.
I use? Yeah, okay, so here I showed the results using the TFFMs. I've done the exact same analysis using classical PWMs, using trained PWMs with different models, and every single time I see I observe the same thing.
Okay, thanks.
So it's not dependent at all on how you call the potential TFBS. It's not dependent on that, for sure.
Okay, thank you.
So I have a quick question. So when you calculate the accuracy for your Ensemble classifier, what was the background that you used? Is it the entire genome or just a small subset of the rest of the genome?
So what I've done is taking from the mappable region of the genome a subset which is matching in terms of length of the foreground and matching the GC composition. I'm currently also looking at matching the dinucleotide composition.
Okay, but it's the same number of...
exact same number, yeah, it's totally balanced. Yes, same length, same number, same GC composition.
Sure, thanks.